A research team has been travelling through the country to try and find 3 grass species that could be significant in long-term efforts to alleviate expected adverse climatological conditions due to climate change. The project is in collaboration between members of 2 universities. Isaac Hofmeyer spoke to Dr. Rose Marks from the University of Illinois and Dr. Llewelyn van der Pas from the University of Cape Town.
So we're studying resurrection grasses, which are grasses that can survive without any water. And we're looking at 3 specific species and trying to understand the natural diversity of them across the entire country.
Okay, so one of them is Eregrostis nadensis. Another one is Oripetium capense. And our favorite one, which we call Chloe, is
Microcloacapra.
It's very similar to what we see in many of the seeds that you can store. So it's similar processes where, when seeds mature, they dry out and they actually become desiccated. These plants kind of do a very similar thing. Obviously, it's very different because we're talking about leaves versus seeds, but it's a very similar parallel system.
This is one of our wettest sites, so we know these species occur in extremely arid regions up all the way into Namibia in the desert.
Yeah.
But they also occur in these wet regions like the Drakensberg, and we're trying to understand if they've lost some of their hardiness in these wetter regions or if they've gained some in the drier regions, and if we can leverage that to breed more drought-tolerant crops or engineer more drought-tolerant crops. So we'll look at the cellular genetic mechanisms of how these plants can survive in a fully desiccated state and try to pull apart the mechanism so that we can improve our crops and our storage system.
So we have some idea that maybe there are certain fungi and bacteria that have perhaps co-evolved with these hardy plants, and we want to explore whether or not there's a particular bacteria or fungi that we could potentially use from these plants to maybe boost productivity for other cereal crops, so like maize and wheats. So it makes for a nice system because we can do comparisons of both above and below ground, and we're quite excited to see what we're going to get. We think that Oripetium and Eregrostis, they tend to like it a bit drier. So what we found is Northern Cape, Kimberley, Bloem, we found more of them there because it tends to be a lot drier there. And we tend to find Chloe in more of the wet areas. So we're not expecting to find them here. We're hoping, we're praying, but we're not expecting to find them here.
Well, there's quite a few more steps of the process. So we've got to bring the plants all back to the greenhouse and cultivate them and get the seed. We'll grow them up in a common garden or in a single space, and we'll do a lot of detailed phenotyping to measure how well they tolerate drought, and then a whole bunch of genetics and genomics to figure out what genes they're expressing and what genetic variation they have that might contribute to increasing or decreasing their resilience.
And for the bugs, it's just a lot of culturing. So a lot of plating onto different media, trying to isolate unique bugs. We're also looking at doing DNA sequencing to identify what's actually which bugs are there. Because if we know what's there, we can maybe try and find a way to culture them. If we can culture them, we can then use them as what we call an inoculum on seeds or plants to see if we can make maize or wheat or even something that's hardy like teff a bit hardier, all within that general umbrella of climate change and possibly ensuring future food security.
With some species that we work with, it's a lot harder. So the grasses tend to be pretty good growers, so the seeds germinate well, they can grow in a lot of different soil types, but surely they're different than in the native environment. But some of the other plants we work with are very hard to culture.
What we try to do, if I'm not lazy, is process the samples as quickly as possible, and then we flash freeze them in liquid nitrogen. So we kind of like preserve what's there. We stop them right there. So when we were collecting from the dry areas, it was fine because we don't expect much to change, with putting them in bags or whatever. But from the wetter areas, we do expect them to change. So the quicker we can process them, I can process them, the better. Research takes a while. We need to convince graduate students to do the work. We like to think of the projects and the big ideas and have someone else do the work.
We like to do parts of the work. The
Yeah.
field work's pretty fun.
Yeah.
I mean, realistically speaking, we could expect to see a publication from this work just describing the diversity in a couple years.
Yeah.
Probably take 1 to 2 years to do all the rest of the downstream experiments. But in terms of finding a key gene and improving maize to be desiccation tolerant, you're talking about a career's worth.
Yeah.
Okay.
It takes a while. It takes a while.
Yeah.
Yeah.
With many challenges facing agriculture these days, no farmer can afford animals that do not add value to the farm. Able to excel in arid and subtropical areas, with its natural disease resistance and environmental tolerance, the Bonsmara is productive where most other breeds would fail. Bonsmara, the breed for all reasons.